Hello, ladies and gentlemen, and welcome to today's broadcast on microbial testing for the pharmaceutical industry, a straightforward path towards regulatory compliance, data integrity, and computer system validation by Darryl, León, and Sandi. I'm Ben Halen of Thermo Fisher Scientific, and I'll be moderating today's event. Before we begin, I'd like to encourage you to engage with us. You can submit as many questions as you want at any time you want during the presentation. Simply type them into the Ask a Question box on the left and click Send. We'll answer as many questions as we have time for at the end of this presentation. You can also join us for our Ask the Expert sessions right after this event. Please welcome Darryl and Sandi.
Thank you, Ben. Hello, everyone. My name is Darryl León, and I'm a Senior Product Manager in Pharma Analytics at Thermo Fisher Scientific. Today, Sandi and I can discuss how Thermo Fisher Scientific can enable you to comply to the computer systems validations for microbial testing in your bioproduction workflow. In our presentation today, we will cover three main areas that may be of interest to anyone needing to comply with microbial identification requirements of their local governmental agency. First, we will provide an overview of the regulatory guidance around validation specifically related to microbial identification in a GMP environment. Second, we will share our real-world examples of how some customers had to learn the hard way what not to do when trying to meet regulatory compliance when dealing with computer systems validation. And finally, we will review how Thermo Fisher Scientific can provide you a complete microbial identification system and solution to meet your data integrity requirements. For overall pharma applications, we have industry-leading capabilities across the entire bioprocessing workflow. We provide integrated solutions across bioprocessing workflows from preclinical through commercial manufacturing. There are two main ways we provide safety and quality: DNA sequencing and real-time PCR technologies. Specifically, we provide high-throughput, highly specific analytical solutions for mycoplasma detection, residual DNA, and virus detection with real-time PCR. And with our DNA sequencing technology, we enable you to do microbial identification of bacterial and fungal organisms. Due to the possible harmful effects of microorganisms, regulatory agencies expect pharmaceutical organizations to have an environmental monitoring program as part of good manufacturing practices. It is also recommended to identify microorganisms at the species level at frequent intervals to establish a valid, current database of contaminants present in a facility. After choosing the ID technology to be implemented, the system needs to be validated and verified. USP 1113 describes the recommendation for verification of ID method. Specifically, the USP monograph recommended to include one of the following tests. Use an existing system to do comparability tests of isolates from routine testing. The number of isolates can be as high as 50. Or testing by the use of, let's say, 12 to 15 reference stock cultures of different commonly isolated species, or confirming 20 to 50 organisms that get ID'd, which includes 50 to 20 species with a reference lab. It is important to note that the parameters used to verify the methods are accurate and reproducible. How the data records are maintained can be based on four main characteristics. These include reliability, accuracy, usability, integrity. Because data integrity is so important, what is data integrity? It is really comprised of several topics. First, there is the focus on the accuracy of data being generated and reported. Next, how the data is being recorded and stored. And finally, data integrity should be managed and processed based on good documentation practices. Now, there are three main ways to achieve compliant data integrity through CSV. First, you can choose to work with a third-party consultant, but you will need to consider their experience with regulatory requirements and quality. Second, you may rely on internal company resources, but they may lack the experience with the MicroSEQ ID system. And finally, you can rely on Thermo Fisher Scientific to have the experience of your MicroSEQ ID system and knowledge of regulatory concerns to assist you with your validation. To expand on this topic, allow me to introduce my colleague Sandi. She has many years of experience in the field working with customers just like you and hearing about their challenges with computer systems validation for microbial identification.
Thanks, Darryl. As you noted, I've had many years' experience helping customers with their computer system validation as they implement environmental monitoring for microbial pathogen identification. As a staff scientist with our field application group, it's my honor to assist customers in this process. And what I'd like to do today is to go through a couple examples of real-world experiences where customers have used either third-party groups to help with their computer system validation, or they've used internal resources to go through this, and to explain how both of these situations have resulted in different experiences for the customer, and the ways that Thermo Fisher has been able to assist them in both. So what I'd like to start with here is the example of a customer that I worked with in years past that used a third-party contractor for their CSV. In this situation, the customer chose to use a third-party contractor because they had multiple sites across North America where they were implementing the CSV at each of these sites. But in each of these cases, the contractor was in control of the installation and access to the MicroSEQ ID system. And the contractor didn't have any training on our system at all. So the users of the system were forced to take training before the contractor had finished their validation process. And then the system was not enabled and not put into use before the validation was complete. So the users were left without a system that they could use, but had been trained on. Now, when this happened, the contractor set out a number of worst case scenarios. And this is pretty typical in any CSV situation. They're looking to test the system to make sure that they enable it to pass any risk that might take place. So this ended up resulting in software conflicts and errors because the contractor didn't know the capabilities of our MicroSEQ ID software. And this resulted in the contractor needing to come back to Thermo Fisher in order to seek resolution for these errors. So this resulted in a lot of time and support being spent not only on my time, but also on the part of our engineering team to go in and to actually repair some of the errors that were taking place and were set up by the use of the contractor setting out these worst case scenarios. In the end, system validation took well over 18 months from the time the system was installed until the time the actual users could start using it for their microbial identification. There were repeated software conflicts and errors, and these took long resolution times because the contractor tried to solve them themselves before they came to me for support. Then the users had to purchase an additional training, and this ended up costing them time and money because they had been trained too early in the process. But the biggest cost then to the user actually ended up being money that they spent having to outsource testing. Because it spent so long in the validation process, they actually weren't able to get their system up and running very quickly, and they had to outsource this environmental testing to others, and that cost them a lot of money. So this was the difficulty they had in working with a third-party contractor. And this wasn't unusual, not for this site and not for other sites where they implemented this particular process. But I'd like to explain a second situation that we had. And this was for a customer that used their own internal resources. Because some of our smaller size customers will take and they'll use their internal IT resources in order to save money. They don't have the money to go to an outside third-party contractor. So they take what resources they have internally and they use them to implement their computer system validation because their internal resources know their computer systems and can network things to meet their internal needs. The difficulty that we have here is that internal IT departments, being responsible for data integration and data storage, they're responsible for all the computer systems across their entire company. So they're not specialists and they aren't aware of risks that need to be based on specific technologies and specific processes. They, again, don't know the MicroSEQ ID system. They can't be experts on everything. So in this case, one size doesn't fit all. And unfortunately for this particular customer, when the FDA came in and audited their facility on their regular annual basis, they found that there were data integrity and data security risks that were unique to the MicroSEQ ID system. In this case, it was the ability to delete and to alter data. And this is a huge risk within a GMP facility. Data backup was not compliant and secure with current federal regulations under the FDA. And there were individual user logins that also weren't compliant. And this resulted in an FDA finding at their audit. This puts the customer at great risk. When you're under an FDA finding, now you're under a timeline in order to get this fixed. The FDA is going to put you under additional scrutiny until they see that you've resolved this issue. The company came to us in this case. They sought manufacturer validation support, and what this allowed them to do then was minimize their system downtime so that they could get back up and running and realize computer system validation that would put them back in a compliant state for their FDA. So this allowed them also to minimize additional dollar spending of sending out samples, because while their system was undergoing their validation, they did have to take it offline internally, and they did have to send samples out to another site in order to be analyzed. So this realized a temporary increase in their turnaround time while these samples were being sent out, but it was minimized compared to what it might have been had they tried to fix it on their own. So these two scenarios here show you what can happen when customers try to either take a third-party contractor or do it on their own. But what they don't realize sometimes is that Thermo Fisher offers one source for a seamless implementation toward regulatory compliance. And we've developed this for our MicroSEQ identification solution. So we bring in the technology and support, starting with the purchase of the system. Our customer buys an instrument, but they also buy the consulting service. We go through the installation and the training. This timeline depends on the customer's availability. If the customer needs things to move more rapidly, we can mobilize our resources to meet that kind of timeline for them. Then we move into our computer system validation phase. Now, yes, it can take 10 to 12 weeks from start to completion, but again, this can depend on customer availability. But we offer them complete project management, documentation, and approvals. We offer them validation, test execution, and customer approvals. We have computer system validation specialists to execute these test plans and to give this information to the customer so that when the customer has implementation of their system, they've been covered from the beginning to the end. What it really comes down to for us is this entire MSID implementation program, where we put the customer at the heart of everything we do. And so it's a holistic approach. And as a field application scientist, we're just part of this circle that is going to be for the customer. So we have detailed training that's there, not just on the assay that comes into play for the bacterial and fungal identification, but we have implementation guidance. Everything that starts with setting up the lab according to your good manufacturing process and your good laboratory and molecular biology practices, but a checklist of implementation materials that would be required. We have introductions to the basics of the DNA-based sequencing and traditional culture methods. We have validation guidance that we're going to give to the customer that helps them plan out how they're going to put their system into a validated state. We have organizations and methods that we can teach the customer so that they can handle everything from low throughput to high throughput. And we have user group workshops where we keep the customer in touch with long-term training, because part of our implementation program is being with the customer from the very first time they contact us about a sale through the very last time they need to contact us for support. Our support doesn't end. We're there for software training. We're there for analysis from data collection through identification. We talk with them about what good acceptance criteria is and interpretation of the sequencing ID results. We talk with them about routine system maintenance so that they know the best practices of their instrument and of their software. We talk with them about data archiving, and we talk with them about recommended performance qualifications. And that's all before we get to the computer system validation. So, Darryl, really our entire program is about making the customer successful. And that's really what I want to get across to everyone. But what else do we have in MicroSEQ and microbial testing that you can tell the customer about?
Thanks, Sandi. I appreciate those user stories, and I agree. There are other things that we need to consider for a microbial identification system. So, in choosing which identification method or technology to adopt, there are really three main parameters to consider. These three main factors to consider are data quality, which is essential for any regulated laboratory environment. There's technology, which is thinking about time to results and thinking about throughput. And I guess like everyone, it's about cost. Basically looking into return on investment. Should you ID in-house or should you outsource? So these are the considerations that one must have. Now, in terms of microbial identification, MicroSEQ ID in particular is a genotypic method based on the sequence analysis of ribosomal genes in bacteria and fungi. The MicroSEQ ID method is a well-established genotypic system that has been adopted by 23 of the top 25 pharmaceutical organizations globally, and 20 out of the top 25 pharmaceuticals have multi-site adoption. So the genetic sequencing has been considered really as the gold standard for phylogenetic classification of bacteria and fungi, and has been designed to support the recommended qualification guidances. Now, the MicroSEQ ID system is a high-throughput comparative DNA sequencing for identification of bacteria, fungi, and yeast. And the whole system includes the PCR and sequencing kits, genetic analyzers or sequencer, and the MicroSEQ ID software and the associated libraries, and the PCR thermocycler. So, one of the solutions is the system, and what it is that one example here is the 3500 XL genetic analyzer, which is really an excellent choice for high throughput labs, shown on the lower side. If you want a more easy to use lower throughput system, this is something you'd use for smaller labs, and we got you covered as well. This system uses the Seq Studio genetic analyzer and uses the same kits and software as the high throughput system that's shown below. Now, the MicroSEQ ID system allows you to either use current species libraries, or you can actually build your own library if you want. In either configuration, the system allows you to choose the right library for your microbial identification projects. And in addition, the software provides you with an SAE compliant reports, auto analysis from sequencing to final reports. Now, our CSV service can assist you when you're preparing for validation as well. We can provide a dedicated staff. We have knowledge of validation, as you heard earlier from Sandi, documentation, SAE, and specifically we can bring really a dedicated project management experience, validation specialist, as mentioned earlier, SAE software workflow, and basically we have a lot of experienced specialists. Now, why choose us? Well, we can really provide complete transparency to facilitate regulatory compliance. That's something we can stand behind as Thermo Fisher Scientific. We have an integrated support team of highly experienced specialists, and our teams are flexible, really, as Sandi mentioned, to really meet your timeline and your business requirements. And finally, you are supported by a whole range of Thermo Fisher Scientific colleagues who can provide the answers you need to move forward in compliance. Simply put, we can save you time, money, and compliance headaches because we can provide you the experience and expertise to meet your microbial identification goals, obviously within a regulatory compliance environment. In terms of computer system validation, we can provide you up to 12 separate components related to consulting services or documentation to help you achieve or maintain your compliance requirements. For example, we can provide you a validation plan, test plans, installation qualifications, quality assurance, all types of things. But to really illustrate how we can do this, I'm going to bring Sandi back, and she can discuss how Thermo Fisher Scientific has helped a customer with their CSV requirements.
Thanks, Darryl. You know, I love what you said earlier about having a team there to support the customer, because this next customer illustration really demonstrates how our teams can come together to give the customer the best CSV experience possible. This was a really unique situation that we had. A longtime customer came to us and asked us to implement a CSV for them, and they gave us a timeline. They said, we need a CSV completed in 6 weeks. So when they meant 6 weeks, they meant 6 weeks turnaround time. And that meant from the time the instrument was installed until the time it was up and running and validated for use. 6 weeks complete. And the new system had to be identical to a current system they had in place in their laboratory. But this would also include updating and expanding their current microbial identification libraries. And we would also then have to follow on very quickly by updating their current system with those new microbial identification libraries. So this was going to take a team if we were going to execute this in the timeline that the customer needed. So this required coordination of all of our specialists, our technical sales specialists, TSS, our field service engineers, FSEs, of course, myself, the FAS, and our compliance team. We all had to get together. And so we had coordinated phone conference calls with the customer and without the customer every week. We took preloaded documents. We took the documents that you just went through in your last slide, Darryl, and we used the ones that had been completed for the previous instrument that we loaded for the customer and set those up for the new system. We prepared a specialized plan for how we could validate the new library for the customer, and then we put together the tightest schedule we possibly could. And the beauty of it all was that by coordinating this team of specialists across the company, we were able to have the system operational and performing microbial identifications within the 6-week time frame that we needed, and we had that complete CSV package. There was no downtime for the customer, and this is extremely important because it was a very high throughput, thousands of microbial identifications a week. This gave them immediate expansion of capabilities. So it was so important that this team came together, we worked, we executed. The customer was extremely pleased with what we were able to do. Darryl, it was really a great experience to work with everybody in this situation.
Thank you, Sandi. I am really glad you're able to share so many customer stories with us. So really, what I wanted to really conclude with is, again, in summary, there are really 3 main takeaways from our presentation today that we can share, and that is, hopefully you quickly realize that Thermo Fisher Scientific offers a complete solution, including instrumentation, assay, software, and service and support to facilitate regulatory compliance for microbial identification. And the MicroSEQ ID system itself offers high accuracy of microbial identification for the GMP environment. And finally, Thermo Fisher Scientific also offers complete CSV services to assist with meeting regulatory compliance. So with that, I want to thank everyone for their time and attention.
Thank you, Darryl and Sandi, for that very informative presentation. I've been monitoring the chat window here, and I see we've already received some great questions in the chat window. So I'm just going to go ahead in with the first question. So first question, I already have a 3500 sequencing instrument. What would I need to purchase in order to have a MicroSEQ ID CSV solution? Specifically, would I need to buy an additional instrument that is specific for MicroSEQ ID?
Yeah, thanks, Ben. I can actually take that one. Yeah, so you don't need to buy an additional instrument if you already have a 3500 sequencing instrument. Pretty much what you would need to do is buy the appropriate MSID assays. You'd have to buy the associated software and also the CSV service and any other kind of services that are needed. So, so the short answer is no, you would not need to buy a complete new instrument.
Okay, great. All right, the next question I saw came in says, Thermo Fisher Scientific has so many types of salespeople and field people. Who do I contact to find out more about my particular needs regarding CSV? And I also want to know how long it takes to get validated.
You know, that's a really great question, Ben. I'd like to answer that one. We do have a lot of people who you can contact within our system, and sometimes the best one to contact is your field application scientist. We can answer a lot of questions about the amount of time it takes to handle a CSV because as FAS, we are on a lot of those calls. And we can work with the customer as we about details that happen during the CSV process. We can also put the customer into contact with the right person to get more information about quotes on the CSV and what it takes to make those things happen.
Okay, well, it looks like we're out of time. As a reminder, though, to our audience, questions that we didn't have time for today, and also for any of those questions that are submitted during the on-demand period of this webinar after it goes live to the web, those will all be addressed by the email address that you provided at the time of registration. For those that are attending the live event as well, I want to again remind you, there's a great opportunity to attend an Ask the Expert session right after this call. So please, if your questions weren't answered here, please feel free to attend that Ask the Expert session as well. So in summary, thank you again to our speakers, Darryl, León, and Sandi, and to really everyone who participated in this presentation. We hope you enjoy the rest of the event, and be sure to check back frequently for new content and presentations added throughout the year.